of the Boston Protein Design and Modeling Club. So thanks everybody for joining us. There is some news before I introduce our speakers. There's some news. So Chris mentioned this back in December, that we are going to be going under the wing of the Open Molecular Software Foundation, where we can actually start to raise our own funds now. So officially we are part of the OMSF. So if you look them up, it's omsf.io. So we're going to be able to raise our own funds so that we might be able to not only buy food and drinks in the future, but also invite speakers from even outside of Boston and pay for them to come give talks. So keep that in mind if you have anybody that you might think is a great idea to invite to speak in the future. Let me or Chris or Sergey know. Chris couldn't be here today. He's at a meeting. Sergey didn't want to infect anybody because he feels under the weather, so he stayed home, which is a great segue into our topic today. So Sarah and Noor are both in Debbie Marx's group, not too far away from here, across the quad. Sarah's a grad student from MIT, actually, and Noor is a postdoc in Debbie's group from Dalhousie, actually, when you got your PhD at Dalhousie. So they've been doing some really cool stuff that they're going to talk about. I'm not going to belabor it, but just to say that some of their work has implications for hopefully nobody in this room has the virus that they're going to be talking about, but I think many of us were already infected with it. So maybe some of the work that they are going to be talking about is going to be important with getting ahead of the next variants. So I'm very excited to hopefully stamp out this pandemic. Maybe some of the work that we'll hear will help. Okay, so without further ado, looking forward to your talk, and take it away.
Thank you so much for the introduction, and we're really excited to be here to speak with you today. Should you check everything is fine with the audio on Zoom? Are we good? Great. So we'll talk to you today about how we can use the past to predict the future. We'll be doing unsupervised early warning of viral antibody escape in order to both design and test future-proof therapeutics. The COVID pandemic, which we see cases for here, has presented a unique opportunity to study viral evolution. Shown here are the COVID cases over time. And what we can see is that surprisingly, at the very beginning of the pandemic, people said the virus isn't mutating quickly, so any vaccine we get, even one based off the original Wuhan spike, will still work down the line. However, as these kind of waves of the pandemic show, this wasn't the case. These waves actually correspond to new variants that occurred in the pandemic, starting first with single mutation variants like D614G, and then progressing to variants of concern like Alpha, Beta, Delta, and then Omicron, which is still highly mutated and still what we're considering what's going around today, even though what's going around today is as different from the first Omicron as Omicron was from Wuhan to begin with. And so now we're in this kind of like JON.1 wave, which is part of this BA2.86 variant, which is 60 plus mutations away from the original Wuhan spike. And so, no, it's not true that this virus didn't mutate, just the fact that it was in such a large population, even though the mutation rate was slow, we've been able to get these gigantic variants. And so these mutations occur, and meaning that the antibodies you get from a vaccine, those no longer protect you once you get exposed to a new virus. And so we call these variants that make it so those antibodies no longer work for protection, antibody escape variants. So these later variants, especially Omicron, have caused significant antibody escape. We focused our efforts on a specific question. Can machine learning facilitate early and accurate predictions of viral escape from antibodies? This will enable us to do several key things for the pandemic. The first is early warning of high escape variants. If we know in advance that a pandemic variant that might cause the next wave is occurring, that can let us do early public health prevention measures. We can also use such a model to evaluate protection of vaccines and therapeutics not just against what's currently circulating, but against what will be circulating in the future. Finally, we can also use it to actually design the vaccines themselves. Let's take a trip back in time. Can we use the past to predict the future? Shown here again is the COVID cases in the pandemic, and what's been remarkable about this pandemic is all of the sequencing that's occurred in experiments on a massive, unprecedented scale. We're at more than 16 million SARS-CoV-2 sequences, more than 690 spike antibody structures. For all of you who are very familiar with the PDB, and especially people who are doing antibody work, you'll know that spike is vastly overrepresented in the PDB in terms of what we know in terms of its structure. And also just so many neutralization, where you're testing the virus against what antibodies are binding, as well as millions of other experiments are being happening. But we're not going to use any of this information for training. Instead, we're going to go back to the pandemic onset and look at what information was available then and say, can we then use that information to have predicted what happened during the pandemic? So how can we possibly model antibody escape without using pandemic information? So we represent antibody escape, the probability that an individual mutation or a strain escapes antibody binding, as a product of three terms. The first term is accessibility. Is this mutated residue in an area that is actually accessible to antibodies? So you think that's kind of generally on the surface of the spike protein. And so all that needs is a 3D structure of spike. From the very first day you had that sequence, you could have alpha folded it and kind of got what turned out to be a very reasonable structure. But also the very first spike structures experimentally were determined March 2020, so very early into the pandemic. Second, it's not just a mutation that is in a site that antibodies are contacting, but one that's actually dissimilar and disrupt antibody binding. So for that, we look at physicochemical properties like charge and hydrophobicity, which are important for protein-protein interactions, in order to say that, okay, this is a mutation and it's actually dissimilar enough that the antibodies that bound the first version are no longer going to bind the second. And lastly, we look at fitness. Just because a mutation was accessible and it was dissimilar doesn't mean that's a mutation that can actually happen in real life. It can't break the protein. The spike still needs to be infectious. So we want a protein that's still going to fold properly, as well as perform its functions like host receptor binding, which in the case of the SARS-CoV-2 spike protein is binding the receptor ACE2. So together, we call these three components EVEscape, which is a deep learning model of viral escape from antibodies. Now let's take a deep dive into each of the three components, starting with accessibility. How do you know where antibodies bind without prior information? A naive approach could just be considering relative solvent accessibility, RSA. This takes a protein and goes all over the surface and says, where is it that a water molecule could bind? And the thing is, that doesn't distinguish between where you are, like if you're near the bulk of like where the protein mass is, or if you're very highly protruded from the bulk. All it cares is that you're actually on the surface and not buried in the core. So the problem there is that shown here is this COVID spike trimer embedded in the viral membrane. And so you'll say that there's equally high RSA for the receptor binding domain at the very top and a surface accessible, but not very protruded area near the core of the protein. And unfortunately, antibodies tend to bind these protruded regions, and neutralizing antibodies especially, these are antibodies that alone can help prevent infection rather than just bind the protein and help contribute to other protection, actually need to be doing something like physically blocking host receptor binding. So again, this is the ACE2 receptor. However, again, there's going to be equally high RSA for places that are blocking receptor binding as other surface areas. So what we consider instead is a different metric called weighted contact number. This is the sum of the reciprocal squared distance between a residue I and every other residue J. So this takes advantage of the fact that more accessible residues are close to few other residues. So that receptor binding domain that's very highly protruded, if you look at the distance from a residue there to every other residue in the spike, it's going to be close to a lot fewer. And so you can see that the most accessible residues are going to be in these flexible regions where you're not in contact with other residues and you're very protruded from the core of the protein. And so you happen to get the highest accessibility from this metric right at the receptor binding area that contacts the ACE2, but only from the spike 3D structure alone, any knowledge of ACE2 binding or any other pandemic information where you know where antibodies bind, like with no experiment saying this is the most neutralizing region. You just get it from the structure. And so what we use is this negative weighted contact number. We flip the sign so that something that's a higher number is something that's highly accessible. And then we take the maximum across protein conformations. So the spike RBD is both in a closed position where the single RBD is down and an open position where the receptor binding domain is up so that it can actually contact ACE2. And so we take the maximum across these because antibody binds both of these conformations. Let's now take a look at the second component, dissimilarity. So which mutations disrupt antibody binding? There are multiple notions of antibody distance in terms of like the specific residues that we could use. The first set of distance measures we could consider are in terms of physicochemical properties, such as a change in hydrophobicity between the wild type and mutant residue, a change in charge, or a change in size. So these are physical properties that we know are likely to have an impact on protein-protein interactions, because something that originally was binding a negatively charged residue and that switched to a positively charged, that's much less likely to bind than making a change that's a lot more similar. You could also look at commonly used distance measures like BLOSSOM, which is an evolutionary measure of how much a mutation tends to be substituted for another one. However, that's not specifically happening in binding footprints. That's just substitutions anywhere. So you could see that that might not actually be a proper measure of distance when you're considering protein-protein interactions. We can also look at different factors from the model, such as in our case, when we're looking at a VAE, we can look at the latent space of that VAE and look at the L1 distance between the mutated and the wild type residue. So we take a look at how these different notions of dissimilarity relate to actually known data, where we take a look at deep mutational scanning data that measures escape against antibodies. This is done by the Bloom lab. And we say in those sites where there is escape, which residues to which mutations are actually occurring and actually causing escape. So those similar mutations, you would say, even though there is escape at that site, it's less likely to actually disrupt antibody binding, and the dissimilar, you're not. So we split into the escape, not escape mutations within those escape sites, and then we do a point biserial correlation with these different measures. And what we see is that though all of them can sometimes be indicative of where escape is happening, hydrophobicity and charge are the most specific. And so we create our own joint charge hydrophobicity dissimilarity score that we use for EVEscape. Lastly, let's look at our fitness model. One way to create an early predictor of residue fitness is to learn from evolutionary sequences, as I'm sure you're all familiar with. Genomes record billions of experiments of what sequences maintain fitness. So the protein's still going to fold properly, and it's still going to engage in the behaviors that's represented in the sequence, such as host receptor binding. And we'll learn from what mutations are contained within our alignment of sequences to say what mutations are possible during the pandemic. So we can learn from evolution in a variety of ways. The simplest model is, of course, an independent model, where you take a multiple sequence alignment of evolutionary sequences, and you look within a single position and ask what mutations are frequent at that site. And then you do that across the different sites, but never considering the context in which those mutations appear. Slightly more complicated would be a couplings or POTS model, where you can look at pairs of sites and say what pairs of mutations are happening and relate that to what you're likely to see in the future. What we use is a variational autoencoder, EVE. For the VAE, this actually considers the mutations in the context of the full sequences in which they occur by essentially embedding the full sequences. And lastly, you could also use transformers. These would be an alignment-free method, where instead of requiring all the sequences in alignment where one position is what you're looking at, instead it's alignment-free. So you're looking at each sequences individually. And the thing about that is it would allow you to score insertions and deletions. Now, indels aren't very common in the SARS-CoV-2 pandemic, but they do occur. And so while we use the VAE as our base model, we are able to sub in a transformer model, Transcept-EVE, when we do need to be scoring indels. But the main focus is on this is just simple substitutions. So, yes.
Actually, I have the microphone, so we should use it.
Yeah, and anyone's welcome to ask questions throughout.
Yeah, I have a quick question about your previous slide.
Yes.
So, I should have warned you that this group will tend to interrupt you.
No worries,
Okay.
We're happy to have questions.
So for the VAE part, how do you just, if anything in the alignment had a gap, you just get rid of it so that everything has the same length?
Yes.
And there's no gaps in this
Yes.
As whatever your reference sequence is.
Yeah, so we use the Wuhan spike, which is 1,273 positions. And even though there are some indels that occurred during the pandemic that then are built upon after that for new mutations, we just align everything to the one reference and only consider things that are that length.
Okay, thanks.
Any other questions? Yes?
All right, a couple slides back, but I'm curious about the accessibility measure.
Yes.
I'm sure you'll get into this later, but I'm curious how much it matters, given that you also have this similarity to the site where the antibody is binding. But I'm also a little confused about the intuition about how you measure accessibility, because are you normalizing those distances, or are you just looking for little dangly pieces coming off the end, essentially?
So let's go back to a 3-component figure. And so what we're ultimately ending up is a single score for accessibility for a mutation, a single score for dissimilarity, and a single score for fitness. And so the question could originally be why not just do a fitness alone? Why do you even need the other two at all? Some problems with that is that, for one, things that are more similar actually tend to have higher fitness when actually you want dissimilar things in this case. And then why between accessibility and dissimilarity, why you need both. Accessibility is going to be the same score for every mutation in that site, whereas dissimilarity is actually going to change per mutation, but not consider the site that's happening. It's a change, just like if I change an alanine to an arginine, doesn't matter what site it is. So a fitness score is unique per mutation, an accessibility score is unique per site, and a dissimilarity is unique per amino acid change.
Okay, that makes a lot of sense. And then the accessibility score itself is really looking for things that are basically distant from everything else in the protein.
Yeah, so let's look at what the spike structure looks like. So especially in the open configuration, we can see the green RBD being up. The maximal accessibility is something that's both on the surface of the RBD, so it's not buried in the center of that green mass at the top, but is protruded. So you can think the minimum accessibility is something that's literally buried in the core of the trimer. Then if you're on the surface of that core thing but still in the membrane, you'll be a little bit higher. And then as you go up and up and up, you'll be higher and higher with the mutations that are actually on the physically on the surface, as opposed to buried being the highest. So it's a measure that cares between both are you literally buried or are you not, and also are you near the core or are you protruded. And it just captures that both all at once in this simple score, just paying attention to every residue's distance to each other. Yes.
Are you just using a centroid for residue?
Yes. That keeps in track of the side chain as well. It's not just the alpha carbon. Yes.
You average this across different conformations?
Yes. Oh no, we take the maximum across the conformations because.
Oh, we see maximum.
Yes. And these aren't the only conformations that spike go in. It also goes into like a weird post-fusion where there's essentially alpha helices down like the core of the like bulk of the trimer, and it like stretches out and it changes. But that's not a conformation in which antibodies bind. But antibodies bind both of these, and neutralizing antibodies in particular do bind both of these, even though the ones that are receptor blocking are the ones that are binding in the open configuration.
And this generally works if you look at other targets besides the spike protein as well for identifying the antibody binding sites.
Yes, and what helps is that this kind of shape, this could, if I told you this was HIV, like it would look like this too. And if I told you it was flu HA, you would say it looks like this too. Like a lot of these common viruses have like class 1 fusion proteins that are all kind of like this trimer, and the receptor binds near the apex of the protein, and it like generally works across viral proteins. So yes. So back to what we do to train our fitness model. So again, because we're only using pre-pandemic information, we take a look at what evolutionary sequences there are for SARS-CoV-2 spike that's available pre-2020. So there are about 3,000 pre-pandemic coronavirus sequences, ranging from SARS-1 and MERS, which are very close, to seasonal coronaviruses, the kind of common cold that everyone gets, that is technically still a coronavirus. But while very different. And so we can see that predictions trained on an EVE model from this pre-pandemic alignment are predictive of spike protein fitness as measured by expression in a deep mutational scan. So what we show here on the x-axis are the EVE predictions from this pre-pandemic alignment, and on the y-axis is expression. However, as you kind of seen in the picture, spike is a trimer. But what this experiment does, because it's easier to just do a smaller protein, is the receptor binding domain. And so you can see that these two things are highly correlated, but there is an area of mutations that the model says are not fit, and the experiment says are fit. And what we see, these are 11 sites that are highly expressed in the experiment. What we find is that 10 of these 11 sites are where the RBD, the receptor binding domain, contacts the rest of the spike trimer. So the experiment doesn't know that those are sites that are constrained by contacts with the rest of the spike, but the model does know. And so that's where you see some discrepancy. So altogether, these 3 components combine to become EVEscape. However, these 3 components are very modular. You could conceivably jointly model the 3 components to get one combined score where you never at any point have 3 individual scores. However, there's an advantage to learning each of the components separately. So we combine the components by first standardizing each of them. This gets the 3 of them all on the same scale. Hello, is that a question? No, I don't think so. So we've standardized them so they're all on the same scale. We then temperature scale them. So we figure out one temperature for each component that allows you to weigh the components by their relative importance. And we figure out these temperatures by comparing our EVEscape scores for deep mutational scans across a bunch of different viruses. And we want one temperature that consistently generalizes across the different viruses. So we looked at flu and HIV and SARS-CoV-2. And we see that in general, it works best if fitness and accessibility are of equal weight, and dissimilarity is considered only half as much. And we use that. We didn't use pandemic sequences in order to determine this, and it works the same regardless of like what virus you're on. So we've learned this once, and kind of that's what we stick to. However, if you did want to, for instance, generate something in an experiment where you all of a sudden care a whole lot more about accessibility, you could alter the weight. And lastly, to finish combining them, we use the logistic function. So we can get these in terms of probabilities from 0 to 1. So this is a simple framework where you combine fitness protein model predictions with biophysical properties when you have properties that are independent but highly relevant to the task that you want. And so you can think of many use cases where you might want to take your own fitness model and combine it with simple biophysical properties in an independent framework like this, so that you can similarly take fitness but combine it with some other property that you want to get a desired score for a mutation. As a result of keeping the 3 components independent, each can easily be updated individually in the case of either newer models or in pandemic data. So while I had previously described a pre-pandemic model that could have been used at the start of the pandemic, now that we are during the pandemic, you could use pandemic data to individually update the 3 components. So for instance, for accessibility, you could use pandemic antibody epitopes. For dissimilarity, you can use deep mutational scanning data that you have. And another thing that's important for antibodies is glycosylation. And you could add these glycans into your dissimilarity metric. Lastly, with fitness, you could plug in any fitness model you want. So I mentioned we were using Eve, but the model works just as well when we plug in Transceptive that lets you do indels, or really whatever the newest, biggest LLM that you want to plug in there, feel free. And because we're keeping it separate from the other components, it allows you to just like very easily just change this one as you will. And you could also include pandemic sequencing into your alignment to train a better fitness model, or even deep mutational scanning data. So let's show an example of how you could update a component. Glycosylation for HIV, even more so than SARS-CoV-2, was important. Shown here is the HIV envelope protein. As you can see, it's from the top, but it really does look like that same trimer that you see for spike, just from the top angle instead of the side angle, but it's completely covered in glycosylations. So the gray is the protein itself, but the green, orange, and pink is just like N-glycans completely covering the surface. This means that for HIV, antibodies have to actually bind those glycans or find little pockets where there are no glycans in order to actually bind the HIV N protein. And so because glycosylation is so important, what we do is we add a loss of N-glycans. So there's a motif, which is N, and then any amino acid besides proline, and then S or T. And you can just identify anything that would disrupt this motif and say that then you're going to have a lost glycosylation. And say anything that loses you glycosylation, we're going to maximize out that dissimilarity component. And that's the only change that we're going to do. And so when we can compare to some deep mutational scanning data, again, from the Bloom lab, that looks at EVEscape from a handful of HIV antibodies. We weren't yet at 2020 when we can do like many deep mutational scanning experiments at once, so it's just a handful. But even then, we can see EVEscape with glycosylation, which is the bar way at the right, has a higher AUPRC than EVEscape alone, accessibility metrics alone, a fitness model alone, or prior EVEscape models. And so glycosylation does help here. Here we can see AUPRC normalized to like the null distribution of EVEscape. So the number of EVEscape mutations over the total number of mutations tested. And so just by doing a simple change to the one dissimilarity component, you're able to get an increase in performance on picking out HIV EVEscape mutations.
Sarah,
Yeah.
it's probably a very standard thing for machine learning and AUPRC, but it's like, so can you
Yes.
say something?
Yeah, so AUPRC is the area under the precision recall curve. What we use it for, especially in the case of kind of very a mixed kinds of amount of EVEscape data and not EVEscape data. As I said here, we're only capturing a fraction of the true epitopes of HIV because only a handful of antibodies are tested. So with this kind of uneven data, we choose AUPRC as a good metric for it because it does better with this kind of uneven amounts of one label versus the other. And here we just use it as a metric of how much of the like true EVEscape mutations you're pulling out.
What's it normalized to?
It's normalized to the fraction of EVEscape mutations. So because there are, say like you're a protein that's like 200 amino acids long, and there are like maybe 10 different epitopes across the surface, but you've only tested 5 antibodies. So there are many false negatives in this data. So what we want to say is, to what extent are you picking out the true EVEscape mutations relative to the fraction of EVEscape mutations that there actually are? So you normalize it to like, if you were like to randomly guess the mutations in the EVEscape mutation, that would be the same idea as what is the fraction of EVEscape mutations in this data set. That's how we like normalize it. Because you're essentially just missing a lot of data because you're not comprehensively testing all the possible antibodies. So the chance of randomly getting an escape mutation for your model would be the fraction of escape mutations.
So if you're above 1 on this,
You're better.
than
Yes. Yeah. Does that make sense to everyone?
Do you mind explaining a little more about the loss of glycan usage, like how you added in?
Yes. So the sequence, you're not entirely likely to have an N, X, S, or T motif be a glycan, but you're like 95% of the time that's what it's going to be. And so we look for, and we can check that anytime we see an N and then anything but a proline and then either an S or a T, that is where we have glycans. And then we say anytime you lose that N or you lose that S or T, that's going to make the glycan disappear. And so anytime you make a mutation where you're going to make a glycan disappear, we say, now you've maximized out the dissimilarity. So we turn whatever your dissimilarity was from charge and hydrophobicity to the maximum possible dissimilarity score, because that's a large change you're making in terms of like what impact that mutation is happening on antibody binding. Does that make sense?
Wait, before you leave.
Yes.
What is with gly?
With glycosylation.
Oh, glycosylation. I'm thinking about glycosylation. So just to follow. So weighted contact number
Does very
does
well.
pretty well here.
It does.
And if you did weighted contact number with glycosylation, how does that do?
I'm not sure, actually. I think it definitely doesn't do as well as EVEscape, but it's been a while since I looked at what exactly that plot is. Again, because this is HIV as opposed to like SARS-CoV-2, where you kind of extensively covered all the different epitopes, it's like an AUPRC where there's like so many false negatives is like still very difficult to interpret. There's like a bigger difference between EVEscape and like WCN when you actually like capture all the epitopes, not just kind of like the most common ones. But yeah, WCN does like surprisingly well. So another advantage to having 3 separate components is the interpretability. So shown here are frequent variants of concern mutations. So these are VOCs like alpha or omicron and different mutations that make those up. And we show here both their EVEscape score and their fitness component EVE score. So when you can look at the top 10% of these, of all the EVEscape mutations, many of the VOCs fall in there, but not all of them do. And what's interesting is to then investigate what's happening with these mutations that aren't escape mutations, but they're really high EVE scores, really contributing to that fitness. And what we can see here is that they are A222V and T547K. A222V results in an increased RBD opening. So the receptor binding domain is up high, and this lets you kind of be more fit without actually causing more escape. And T547T stabilizes the conformation. So this enhances your ability to transmit, but that also doesn't impact escape. So they're important for infectivity and kind of spreading the virus, but not directly relevant to escape. And so by looking at these 3 components individually, give us an added interpretability than having a joint score alone would. So this leads us to the key question. Can EVEscape, trained on pre-pandemic data, anticipate pandemic mutations? We'll take a trip back in time to evaluate the predictions. I'd mentioned to you earlier just how much data is out there. All of the sequencing data, so many structures, but we're not using any of this for model training. That means all of it is available to evaluate our model. So we ask, how well do current methods comprehensively predict future immune escape? So shown here is the fraction of predicted mutations when you look at the top 10% of predictions. What fraction of those are seen in the pandemic? What you would think is almost a ground truth measure of what mutations are going to cause antibody escape is taking pandemic antibodies from patients who have had SARS-CoV-2 and actually doing a deep mutational scan where you make every single mutation and say, does it bind that antibody or not? So you'd say that would tell you a whole lot about what the future EVEscape mutations are, because you're actually doing real testing against pandemic antibodies. Now, of course, that takes 10 months in the pandemic for you to do, just because experiments take a long time, but also you actually need pandemic antibodies. So you need to wait for people to get the relevant infections in order to get the antibodies to test against. So it takes 10 months. You could say, let's look at more updated antibodies, where we now look at many more antibodies that cover a wider range of epitopes, as well as looking at polyclonal patient sera, where it's not monoclonal single antibodies, but actually kind of the full set of sera from a patient. And we can see while the earlier set of antibodies at 10 months gets you a little more than 30%, by including more up-to-date and more antibodies in sera, you get a little bit above 40%. EVEscape from pre-pandemic information alone gets you almost 50%, just as good or better as what you would consider like a real measure of what the antibody escape is. So those experiments actually testing what mutations are going to have effect on actual patient sera and antibodies. So that's a pretty convincing argument that EVEscape is capturing something that's really useful to understand the pandemic. And it's more than like twice as predictive as prior unsupervised models at predicting pandemic mutations. We can also ask, is EVEscape and those experiments, those deep mutational scans, actually predicting the same set of mutations or different sets of escape mutations? So we look at the top 10% of predictions and make this Venn diagram of which ones are in the top 10% of EVEscape, which ones are in the top 10% of these deep mutational scans, and then which of the mutations have or have not been seen in the pandemic. And we can see while there's like relatively equal fractions of both that have been seen in the pandemic, it's a not completely overlapping set. So actually by combining the information from EVEscape with what you're getting from deep mutational scans, you can be even more predictive of pandemic mutations. It's also important to consider not just what are we predicting general pandemic mutations, but are we predicting the very frequent pandemic mutations? So instead of date on the x-axis, now it's looking at the frequency that each mutation has been seen. And then on the y-axis, it's the percent of pandemic mutants that were predicted in the top 10% of the models. And we can see that EVEscape is especially good at predicting the very frequent mutations that are seen more than 1 million times. So these are the ones that are appearing in variants of concern like Omicron. So we'd ask the question, can EVEscape trained on pre-pandemic data anticipate pandemic mutations? So we say from those sides, yes. And what I'm going to show here is actually what it looks like when we look at EVEscape scores on the structure. So shown here is the structure of the trimer of spike, and highlighted is the receptor binding domain at the top, and at the left, the N-terminal domain. And the circles are any sites that have been seen in the pandemic in the GISAID sequencing database more than 10,000 times. And we can see that the red, the highest EVEscape sites, correspond well to the circles, which is what all the plots in the past couple of slides were telling you, that EVEscape does predict pandemic mutations. But more than that, it's predicting the general antigenic regions, the regions where antibodies are binding, without any information that these were the regions that were going to become the neutralizing antigenic regions of the pandemic. So it could tell you way ahead of time that the RBD and the NTD are the antigenic regions for spike. So, yes.
Question on Zoom. So when you say that there's no information that you've used about this stuff, when you trained the logistic regression model, it was on just sequences of other viruses, or was it on other? Did you know where antibodies bound to those other viral proteins? Because there's lots of similar spike proteins to the SARS-CoV-2. I mean, there was the SARS-CoV-1, or I guess, and there's lots of antibodies that bind to that. Was that?
None of that information was in there. So again, the only information in there is a SARS-CoV-2 spike structure for accessibility, the charge hydrophobicity score that we make from just differences in charge and hydrophobicity for dissimilarity, and fitness is purely sequence information from 3,000 prior coronaviruses. No information about knowing where antibodies bound in other viruses or any other information outside of that.
When you train the weights, that sounds
Yeah.
great. When you train the weights of your, what you call the temperatures,
Yes.
that was to what data?
So that's to deep mutational scanning data. So yes, there's that slide information about looking at deep mutational scans for flu, HIV, and SARS-2. The thing is, we're not like saying this is a virus-specific measure. Like these weights work for SARS-2, but they wouldn't work for HIV. All we wanted was one number, so which ended up being 1 for accessibility, 1 for fitness, and 1/2 for dissimilarity that we say is generally applicable across viruses and it never gets changed. So not trained on pandemic, but is trained on that little bit of escape mutation data just to give you that single number. But we could have just done it on flu and HIV alone, and we still would have gotten that same number. But we do show that you also could use those same weights for SARS-CoV-2.
Okay, so the weights here were trained on data from flu and from HIV.
And we did check on that weight works for SARS-CoV-2 too, but it really is just that single number to say, okay, let's weight dissimilarity half as much as accessibility and fitness.
Okay, thanks.
Yes.
So because the fitness measures come from a whole bunch of related viruses that are previous, theoretically, right, those databases could be full of different variants of that virus that have evolved because that's where antibodies found them, right?
Yes, that's true.
Are you concerned about that? Or is that, as if you look at previous coronaviruses, for instance, that's not really a concern because you just don't have that many sequences.
So, for instance, if we consider what's in our alignment for SARS-CoV-2, it's a lot of bat and porcine coronaviruses. Technically, those viruses are also under selection, and some of that might be due to antibodies, though antibodies aren't really important for bats in the same way that they are for humans. So yes, we're not saying that there isn't some evidence of antigenic selectness in a fitness score alone, but it certainly wasn't enough. It's not like escape was the primary driver of fitness, such to the point that we wouldn't even need an escape, a dissimilarity measure or an accessibility measure. But yeah, there's certainly, I'm sure, some signal in there of terms of where antibodies bound, though we're not explicitly getting any sort of antibody information.
Yeah, makes sense.
Yep.
Just to so I understood well, you need a structure and then you need a bunch of started sequences
Yes.
to put your model. So hypothetically, if you take the sequences in the middle of pandemic, not from the start of the pandemic, let's say the sequences just on the year 2021.
Yes.
So the model still works just on the.
Yes, and actually it's a better predictor of the upcoming years. So if we're considering how well you're predicting fitness, as we say, if I could I could train up through 2020 information and then try and predict what's going to happen in 2021 and onwards, something that has a little bit of pandemic information will continue to do better and better, because it will be able to kind of better capture the exact epistasis that's happening based off having some pandemic sequences. So
Even if you know the sequence.
So you use both the evolutionary sequence information and the pandemic sequences. Of course, then it's a challenge of how to weight the pandemic sequences, but because there can be millions of them, but it's only a handful of mutations, but it is local, highly relevant information. So it's how do you weight those so you don't completely collapse them away, because you still want to consider what's happening in the broader evolutionary space, but still learn that information. And we kind of show that, yes, by using the information after 2021, then we could then have that newer trained version of our model that uses some amount of pandemic sequences and predict what's going on later. And actually some of the results that Noor is going to show is using some amount of pandemic sequencing in your model, where it's you use information up through the alpha variant and then use that to predict what's happening after that, or use information up to the Omicron variant.
The interesting thing though is if you train on just the SARS-CoV-2 data, so just the pandemic data, the model doesn't do as well as this pre-pandemic model. So it's the evolutionary information does become important in this case.
There's a question on Zoom, so I
Great.
figure this is a good time to ask it. The question is, let's see, what data is used to check against the predicted probability of escape?
So we compare to two things. One is comparing to the deep mutational scans alone. So I mentioned like for fitness, you can compare to deep mutational scans of expression, but there's also deep mutational scans of escape. So it's binding to antibodies or patient sera. And so we can compare directly our EVEscape predictions against that, but then how do you put that result into context? And also the things that are tested is not a fully complete, fully accurate. Experiments never are, a model never is. What's the true importance is that you can predict what happened in the pandemic. So we ask, how well does EVEscape predict the pandemic? And how well do the deep mutational scans predict the pandemic? And showed that EVEscape was just as good or even better as those deep mutational scans on patient antibodies at predicting what happened in the pandemic. And I think we left it at just the right moment to move it over to Noor to talk about how we can now apply EVEscape.
Okay, it's working. Thank you, Sarah. So I think this talk's a bit cursed because every time we're about to give it, I get sick. So my voice might come in and out. So just forgive me for that. And this will be the last time we give this talk. No, I'm joking. I think it's because we were talking about SARS-CoV-2. Okay, so Sarah has hopefully convinced you guys that we have this EVEscape model that uses evolutionary information and biophysical information, and that it can predict pandemic mutations. Now, what can we do with this model now that we have it? How can we use it in this current pandemic, but also for future viral outbreaks that are going to probably become more and more common? So we've thought of 3 ways that we can use it, but if you have any other suggestions or ideas, let us know. The first is as an early warning system for high escape variants. So the pandemic is still with us, it hasn't gone anywhere, and there's still SARS-CoV-2 infections going on. And the idea is, can we survey all the sequences that are coming and being deposited online and identify what the next Omicron is going to be? Another application is, how can we use this model as a way to evaluate how well vaccines and therapeutics work, not just against the sequences that are existing now, but how well they might work against future variants that might arise in the future. And then the last application is actually as a method to design vaccines. So a lot of us here probably got the mRNA vaccine, where it had the spike sequence from the original Wuhan strain. But a year or so later, we then were recommended the bivalent booster vaccine that had the XBB variant and the original Wuhan strain. So the idea is, could you have designed a spike sequence from the very beginning that would have made you have the immune response you needed to protect you from the variants that were around then, but also the variants that have come later on and are continuing to arise? So first, as an early warning system. So as Sarah showed in the very beginning of this talk, we've had these waves of SARS-CoV-2 infections and all these different variants. So you have a variant that arises like the Alpha variant earlier on, and then it slowly takes over and becomes the dominant strain globally. And then we had the Delta variant, which I seem to remember because it was like the big one right as the vaccines were being rolled out. And then of course, Omicron, which was just a disaster. And then we now are living with all the Omicron descendants, which if you haven't been around the SARS-CoV or are basically sick of SARS-CoV-2, maybe you don't keep up with it, but they're called all these different alphabet, BQ.1, XBB, CH.1, BA.2.86. These are all sort of descendants of Omicrons that have continued to evolve. So ideally, what one would be able to do is when one of these variants, before it takes over, it has to start with 1 or 2 sequences. And can we identify from the first 2 sequences that this is going to be the variant that's going to take over, so that we can implement some public health measures to say, we identified this variant in this country, it looks like it's going to be a problem, maybe introduce some masking requirements, travel restrictions, work from home suggestions, things of that nature. The problem with that, and why that's hasn't been possible, is that throughout the pandemic, there's been about 400,000 SARS-CoV-2 genomes that are being deposited every month. And that number is just impossible to fully characterize experimentally. No one's ever going to sit there and assay 400,000 sequences of anything, probably. Also, you see that this number is going down. I sadly don't think that that's because there's fewer SARS-CoV-2 infections. It's probably just because we're not sequencing them as much here in the US, but also globally. So the question is, can we sort of find that needle in the haystack? Can we find the variant that's going to be the next Omicron, that's going to be the next variant that takes over the globe, and can we do it early? So what we've been doing for the last year and a half, 2 years, is every 2 weeks, we survey all the sequences that get deposited. So luckily with SARS-CoV-2, there's been amazing data sharing where whenever a SARS-CoV-2 genome, someone gets an infection, they get sequenced, it gets posted online, and we have over 60 million genomes that are publicly available. And that happens daily. Those sequences are updated daily. So we look at the sequences that are circulating in a 2-week time period, and then we identify these variants up here that have the high escape variants, the ones that we think are going to be problematic. And we basically post that information publicly on our EVEscape website, but we also share it with our funders, CEPI, and then they share it with the CDC and the European CDC and so on. And sort of to make them trust us, and maybe to make you trust us also, you could see that variants like CH.1.1, which was an Omicron variant that really took over, is one of the highest. If you look at when it first arose with just one sequence, we predicted that it has the highest escape score out of all the sequences that were circulating within that 2-week time period. And it wasn't just a one-time incident. This has happened repeatedly throughout the pandemic. If you look at the beta variant, which was very, very early on, if you look at when it first arose, when one sequence was deposited, if you compare it to the sequences of everything circulating at that time, it was predicted to have the highest escape. And that's the same for the first Omicron variant, BA.1, and then these later Omicron variants. Okay, so we wanted to go beyond just surveillance. We were thinking, okay, well, we have gotten all these vaccines, we're recommended more booster vaccines. Is there a way where we can use this approach to evaluate vaccines? And the traditional way that vaccines are evaluated is against existing variants. So for example, when the XBB booster or the other bivalent boosters were being rolled out, they were assessed on their ability to protect against the variants that were circulating at the time. But ideally, you'd want some way to not only evaluate against that, but to also evaluate against the variants that might arise into the future. But this, of course, has been historically difficult because we haven't had EVEscape. And also because if you imagine, one could say, well, you might not need it. What if you just take the protein and randomly mutate it? Some might be functional and you could use those. The challenge with that I'm sure a lot of people in protein design and engineering appreciate is that if you take a protein and you introduce mutations at random, the vast majority of them are going to be non-functional, especially if you go further away from whatever wild type you're starting with. So this example that we're looking at here, so the deep mutational scanning experiments that Sarah was referring to in the Bloom's lab, they take, say, for example, the receptor binding domain, and they introduce mutations at random using error-prone PCR. So you can see here that variants that are one mutation away, about 70% of them are functional, but as you move to things that have more than 8 mutations, the proportion of functional sequences drops quite a bit to less than 2%. So you can't use this approach to randomly mutate sequences and identify variants that have multiple mutations. The Bloom lab also did something a bit different, where they were like, okay, we have 3 years essentially of pandemic data. What if we look at that and see mutations that are frequent during the pandemic, so we know they're not deleterious individually, and we do combinations of these individually non-deleterious mutations. Even with that, where each individual is fine on its own, when you do the combinations, you see this drastic decrease in functionality as you move further away from the wild type. So, yeah.
If the these are known mutations that are supposed to be good, why is the number of mutations 1? Why is that not a proportion of viable sequences? Why is that not 1? Why is that?
Well, they don't know. They saw the mutations in combinations in the pandemic. So that mutation could have arose with another compensatory mutation next to it. So they sort of just took the singles. Okay. So what we thought is, what if we could design multi-mutant proteins that are functional using EVEscape? So we start with the EVEscape framework, and we score all possible single mutations, which is about 120,000 for the full spike. And we take the top 1% of highest EVEscape score mutations. We then score all possible doubles, which is about 140,000 sequences. And then we combine those high scoring doubles to make higher order mutants that have 4, 6, up to 10 mutations away from whatever the variant is that we're designing on. We take those variants, and then with collaborators at UMass and the Brigham and Women's Hospital, we test if they're infectious, and we put them in pseudoviruses, and then we test if they're infectious and whether they're neutralized or not from sera that we got. I mean, luckily being in Boston, we have access to amazing sera or blood, like from patients that have had SARS-CoV-2 infections throughout the pandemic. And we see whether that variant is neutralized by someone who had an XBB infection or someone who had a booster vaccination. In total, we designed 83 multi-mutant spike proteins that have about up to 10 mutations away from whatever the base variant variant that it was designed on. Okay, so you see that in our designs, we have a much higher success rate than if you were to introduce mutations randomly or if you were to combine individual mutations that are fine. We had a success rate of about 94%. But the question is, do these design proteins really mimic future viral evolution? Are they useful for our task, which is we want to see if we can predict the future so that we can evaluate vaccines early. And in order to do that, these designs have to really foreshadow the future, or else it's a bit useless. So one way of doing that is to look at an antigenic cartography. And just to give you an intuition of what this graph is, each point is going to be a variant. And that variant has a neutralization score across different immunization histories, whether someone had had a convalescent infection very early on in the pandemic, or if someone had one vaccine or 2 or 3 or 4, whether they had an XBB infection and so on. So we have all these different, I think in total we have over 30 sera panels that we compare against. But we reduce that information into a 2-dimensional map, which is plotted here. So the closer 2 points are in this map, the more similar they are antigenically. And the more further away they are, it means they're more antigenically different. So for example, if you look at just the variants of concern, which are plotted here, and they're colored based on when they arose during the pandemic, you see that the earlier Wuhan variant is right here, and clustering around it are the alpha, beta, delta variants that came very early on in the pandemic. Then a bit further away, you see the early Omicrons here, and then the later Omicrons furthest away in these darker red here. Yeah.
What are the axes? They're labeled to be identical, so
Mhm.
it should be so it must be different than axes.
It's essentially like a PCA. It's two components of PCA.
Component 1 and slope component 2.
Right, but that's the same intuition. I think it's actually a multidimensional scaling. So we have like a 10-dimensional vector that we reduce down to 2 dimensions, trying to maximize the keep the distance consistent.
It's confusing because the axes are labeled exactly.
Mm-hmm.
So I just wanted to know what it is.
Okay, so one thing that's interesting to note is here you have SARS-1 down at the corner there, and you'll see that some of the later Omicrons are as far away from the original Wuhan strain as SARS-1. So we're really entering this new world of SARS-CoV-2. Debbie Marks always says, at what point are we going to start calling this SARS-CoV-3? It's getting to a point where it's quite different than what started 3 years ago. Okay, so now we're going to show you the designs, which are in triangles. So you see that if we took the...
Just because it's more distant doesn't mean it's closer to SARS-1.
Right.
So your principal component is the distance to the initial.
Yeah, and to each other. It's not just to the initial.
So those get close and then
Yeah.
you get cross-reactivity of SARS-1.
Oh, yeah, for sure. For sure. Yeah. Okay, so now we're showing the designs and the triangles, and you can see that when you take, so what we do here is we take the model that Sarah described and we add all the pandemic sequences up until when a variant arose, in this case, BA.2.12.1, and then we design on that. And you see the designs on the triangle, and you can see them moving towards the actual variants that arose later on, this BA.4.5 variant. We then did that again on the BA.4.5 variant, where we took that, all the data up until that point, and designed on that. And you can see that that moves you towards the BQ.1 variants, which came about 8 months later in the pandemic. You do that again on the BA.2.7.5 background, and you see that moves you towards the XBB variants that happened 9 months later. And if you keep going, even on the XBB variants, we're getting towards the CH.1.1 variant, which happened 3 months later. So just a quick recap, we've shown that this model can design multi-mutant proteins. Not only that, those proteins tend to foreshadow the type of antigenic evolution that you would have seen in the virus. So perhaps this is might be a useful way to evaluate vaccines early. So the idea here is you have your prior variants that are circulating. You can sort of design variants that give you an idea of what might happen. Or you can wait to see what the later variants really look like. And this, I think, point that we're trying to make here is best illustrated with an example. So if you consider the bivalent booster, at the time when they first approved that vaccine, you look at what variants were circulating at the time, which are shown in green here, and that's how that vaccine was evaluated. But you could have also, at that time, evaluated on the design variants, which are in blue, which might have told you something about how that booster vaccine would have protected you against what really happened, which is in yellow. So if we look at that, we're showing here neutralization titers, which we've been talking about EVEscape, which is the inverse of that. So things that are lower down means you have high escape. The higher up it is, it means it has lower escape. So if you look at people who've been vaccinated with the bivalent booster, and if you see how well their antibodies protect them from the prior variants, that's pretty well. If you look at the designs that would have been available at the time of the bivalent booster, you can see that we would have predicted a level of escape. There's a distribution of predictions here, but some of them are quite a bit lower than what we knew at the time. And then now that this bivalent vaccine has already been rolled out and we know what variants are circulating now, we can see that we would have predicted a similar level of escape to the later variants have similar levels of escape to what we had designed without having any of that information.
For the designed constructs, were those sequences of the virus actually observed, or were the mutations observed in a background of other mutations of the sequences? 'Cause I guess when you say when you're evaluating your designed constructs against all of the data you have of sequenced spike, were there actual, these were the only mutations in spike, or was it there were these mutations plus many more or some others for antibody escape?
Sorry, so you're asking in the designs, were there mutations there that hadn't been seen at all in the pandemic?
Well, I guess what I'm saying is, like, so you say neutralization on the y-axis, and then you have designed constructs. And what I want to know is, are those sequences that were the designed sequences actually observed sequences in patients, or were those mutations were present in those sequences, but there were additional mutations too?
Oh, I see. Okay, so maybe I can clarify. So what we're doing here is we have sera from people who got a bivalent booster vaccine. They were never vaccinated or saw the design sequences. We then make the design sequence, we put it on a pseudovirus, and then we see how well that sera can neutralize that fake virus, essentially.
I see. I thought this was all computational work.
Oh, no, sorry.
I see.
This is wet lab work, yeah.
Okay, got it.
May as well have it. I mean, I get that, right? That predicts future outcome of your existing vaccine. The other question is, could you use your design? Because I think the bivalent was what, BA4 and BA5 plus the original strain.
Yeah.
Could you take your future designs to better predict what a bivalent should be?
Yeah.
Because
I-
that's the right.
Yeah, so that's the next step. That's what we get, actually, good segue. So how do we actually use this to actually design better vaccines? But maybe before I get to that, I think there was one more question.
So for your design sequences, did you test if this can still infect?
Yes. So we tested whether they can infect, and 94% of them were infectious across the different mutational depths. Okay, so now, if there aren't any other questions about that, we'll move on to the vaccine design. So you mentioned one type of vaccine, the mRNA vaccine. We all are sort of familiar with that now because of the last 3 years, but there's also been other vaccines. And as you can imagine, since this pandemic, there's been huge and increasing interest. People are putting millions and millions of dollars into this for specifically designing pan-corona vaccines. So some people are putting money on a single vaccine that will protect you from all the coronaviruses. Whether that's possible or not, we'll see. But these are some examples of them. A lot of them are in preclinical trials. Some of them are in clinical trials. One in particular that we're interested in, which is not an mRNA, it's a nanoparticle-based vaccine, which I was listening to This Week in Virology podcast. If no one listens to it, highly recommend. And they were interviewing Pamela Bjorkman, who's developing this. And as you're hearing, you're like, well, we could design this vaccine for them. But the idea is, instead of an mRNA, you have a nanoparticle, which is a protein, that you can essentially decorate with other viral antigens, whatever antigen you want. In their case, they put on the receptor binding domain, and they have two flavors of the vaccine. They either have the homotypic, where they put on the RBD from a beta variant of SARS-CoV-2, and that's it. Then they have a mosaic RBD where they decorate it with 8 different receptor binding domains from 8 different sarbecoviruses. So they put in bat coronaviruses and other coronaviruses in there as well. And what they showed is that if you vaccinate mice and monkeys with the mosaic nanoparticle, you can get better and broader protection against other coronaviruses, even ones that are not in the vaccine. So if you vaccinate with the mosaic, which doesn't have SARS-1, you get protection from SARS-1, as well as protection from SARS-2 and all the variants of SARS-2. So this is moving forward as a candidate for a pan-corona vaccine right now. And the reason they think this approach works is that because of the nanoparticle, they hypothesize that when an antibody binds, it has to bind with avidity, which means both arms of the antibodies have to bind 2 different RBDs. So what ends up happening is the antibody binds the conserved region in the 2 RBDs. So when you vaccinate with a mosaic nanoparticle, you get antibodies towards the conserved regions, whereas when you vaccinate with a homotypic, which has just one flavor, which is the SARS-CoV-2 beta RBD, you tend to bind these variable regions like the receptor binding motif. So that sounds great, and it's a promising candidate. There's obviously certain caveats and ways that we think we can improve that computationally. For example, most of the elicited antibodies in the mosaic nanoparticle target conserved regions in the RBD, which is great. You want them to target conserved regions because hypothetically, if they've been conserved in the past across broad coronaviruses, they're going to continue to be conserved into the future. The challenge with that is because the mosaic only has the RBD, most of the conserved regions actually end up being regions that contact the rest of spike. So even though they're accessible and can bind in the nanoparticle, in a real infection, when a virus is infecting with the whole spike, not just the receptor binding motif, those regions are no longer accessible. So the antibodies that you would have made can no longer bind and neutralize that virus. So what we thought is, okay, what if we computationally design vaccines that focus antibodies to specific regions that we want them to be focused towards? So if you imagine the nanoparticle here, and you can design 2 different receptor binding motifs, where you have a specific target binding region, that's the region that we want the antibodies to bind. And we have certain criteria for that. 1, it needs to be conserved because or less likely to mutate, so that it's unlikely to easily escape. And 2, it has to be accessible not just in the nanoparticle, but has to be accessible for antibodies to bind in a real infection. So to optimize the antibody binding to that region, what we do is we computationally introduce mutations to the rest of the receptor binding motif and ignore regions that already have sufficient natural diversity. So we've sent them 10 of our nanoparticle vaccines now, which they're computationally testing. Sadly, we don't have any of those results yet, so we don't know if they've worked better than what the natural vaccine or the natural mosaic nanoparticle or not, but hopefully we can update you on that in another talk at some point. So just to summarize.
Quick question. What's the nature of the computational mutations that you've computed? Do you just sample from Eve, from like fitness, or
yeah, it's
cyan regions here? How do you make those mutations?
We identify an average like footprint of where an antibody could bind, and we sort of scan that, we call that like an antibody binding epitope, and we say if you have a specific RBD, we pick a first mutation at random, and then you pick one that's furthest away, like not within that antibody binding epitope, you introduce it there, and you keep going until you have enough diversity in the RBD that there's no region that's similar between two different ones. You basically start in one place, you pick the next furthest place, you add a mutation there, and you keep going until a certain radius is, every certain radius is covered with at least one mutation, ends up being about 10.
Right. I think maybe my question is even simpler than that.
Oh.
Is how do you choose what that mutation is?
Oh, right, using EVEscape.
Okay, so
Yeah.
you have a probability distribution of the 20 amino acids when you define this is with the residue I'm going to mutate.
Exactly, you choose
You
the
sample
residue,
from-
then you sample from
And
the,
EVEscape gives you that probability distribution of the
Exactly.
amino acids?
Yeah.
Okay,
and then you do that many times, and then you have a few sequences, yeah.
got it.
Okay, so Sarah's told you about EVEscape, and we've talked about a few applications as a surveillance or early warning system, as a way to evaluate vaccines early, and also as a way to design vaccines. The other thing is basically all of this work is applicable to other viruses. So Sarah talked a bit about HIV. We're also doing work on Lassa and Nipah. You've also done some work on flu. So it's virus agnostic and can be applied to any other virus. Okay, so with that, we'd like to thank you for listening, and everyone in the Marks Lab and our collaborators.
All right, I know we have peppered them with a lot of questions throughout the talk, but if anybody has more questions, I can walk around with the microphone. Yep, okay.
So using EVEscape, are you able to predict the course that the pandemic is likely to take from here? For example, if it's likely that there's going to be another wave like Omicron?
Yeah, I mean, Omicron was kind of hard because it was in a chronically infected patient. It's sort of like a bit like being hit by an asteroid. But whether we can predict the next sort of steps in the pandemic, to a certain extent, as Sarah was showing, you can. It's of course hard because evolution is stochastic, and there's going to be some mutations that get fixed just by random chance.
So
Yeah.
to ask differently, we haven't yet saturated the high escape mutations. There are like many more that the virus could explore.
Yeah, in my opinion, I think yes.
And
I don't think we're anywhere. Yeah.
And especially like everything's always escaped relative to something. So we escaped the Wuhan antibodies by our Omicron antibodies, but now we've gotten Omicron antibodies if you are vaccinated or infected. And so now it's new mutations that will escape those antibodies. And so it's kind of like why we keep getting new waves of flu, is that it just keeps mutating away from the protection you do have from some flu variant a while ago. And so we definitely haven't fully saturated the escape potential.
We're also getting a lot of reversion mutations. So mutations that would have been neutralized early on by like the earlier antibodies that we had. And then now we've sort of gone further away, that reverting back to what it looked like before seems to be a good idea for the virus.
One thing, kind of thinking about it, and you may be able to simulate that or can look at it, right? It seems not considering selective pressure is on one end necessary for getting the right predictions, but it also takes away a big component of the path of the pandemic or any sort of viral evolution.
Yeah.
When you played with kind of, you include more of the pandemic sequences, you get some of the selective pressure in. And
So if we include pandemic sequences, you can both like kind of capture epistasis a little bit better, but also it's just like the sequences that are happening at time A are due to selective pressures in the past. The sequences that are going to happen in the future are due to selective pressures that aren't going to be captured in the past sequences. So it kind of keeps changing. However, if we do want to say, let's figure out escape from the specific selective pressure of a specific monoclonal antibody given to patients, we can then look at EVEscape scores within the footprint of that monoclonal and say, this would be the escape that would occur as a result of that therapeutic. Similarly, if like everyone started taking our nanoparticle vaccine, not happening, but you're going to have the selective pressure towards that conserved region that we said is conserved theoretically, but of course some slight mutations are possible, and we'd be able to forecast perhaps a selective pressure that's not happening just independently, but also as a direct result of any specific therapeutic.
it seems like you can look at specific questions, but it's going to be really hard to capture kind of the real core. So
yeah, and I mean one point about like there are a lot of pandemic data. There's 16 million SARS-CoV-2 sequences. They're so similar to each other that the amount of information you're actually getting from them isn't as much as you think, given this big number that we're saying and seeing.
I have a question while I walk to the back of the room, which is you show the plot a lot of the number of counts versus the variant. So it's this, you get these peaks, and then some peaks are very much bigger than other peaks. And I sort of wonder if it's possible to predict the height of those peaks, you know, just how much these variants might take over. As because some of them take over more. There's more cases than others. I'm just curious what you think about that.
So there's a couple kind of different factors for that. I would say the peaks early on aren't actually due to antibody escape. They're due to the fact that this was a virus that was in bats and it's learning how to behave in a human. And so while there is some amount of antibody escape in the case of alpha, what it's really doing is a peak where it learns how to be transmissible to humans. So, but then later on with Omicron, what you kind of get, the peaks might be due to the fact that we all have different backgrounds of like what strains we like have and haven't been exposed to due to vaccines or infections. And something's going to reach perhaps a higher height if it's more dissimilar for a larger portion of the people. So if like we're at the case where everyone has been vaccinated with Omicron, then something has to be significantly different from that. But for like a point, like before Omicron occurred, like Omicron was vastly different from everything that had been seen before because it was in a chronically infected individual that kind of maybe had HIV or something, and so kind of couldn't mount an immune response. And then it was allowed to get like 20 mutations different from anything like further seen in the past. And it was significantly different from every infection that like any individual had seen. And so it has a greater difference on the whole population when it's different from everybody, not just different from like what a small portion of the people's infections have looked like.
Yeah, I mean, it could also just be sequencing.
That's also true.
So we'd have to look at test positivity, how many people are getting tested versus testing positive. But like, I mean, I haven't really appreciated, but just looking at this now, Alpha, Delta, there were much fewer sequencing efforts happening at the time. The smaller peaks here, I mean, BA.2 to 86 has as many mutations to Omicron as Omicron did to Wuhan. So it's really it has over 60 mutations to the Wuhan strain. So even if we don't see a peak that's high, it could just be that we're not sequencing as much anymore.
This is positive tests or sequence?
This is cases. This is number of
positive tests. So
but we don't know how many. That is true. But if you have more, we don't know how many people tested versus how many people got infected.
yeah, and so you probably have a complete underestimation of your peaks.
Exactly.
No one's testing.
Exactly. And maybe at the beginning too.
Can you theoretically predict though, because at every point here you're getting a distribution of actual sequences that you're finding. Can you predict which ones are likely to essentially fix in the human population? Right, because during the early on in the pandemic, especially, weren't there multiple lineages coexisting for a long time? And so you probably might be able to maybe estimate fixation if you assume that you're sampling well. Is that right?
So the COVID is kind of like more different than some other viruses in the fact that, yeah, you get these multiple branches, and then it's never just like one branch takes off and becomes everything, besides kind of the case of Omicron, where it was just so different that it kind of completely took off. But you kind of get this like very like dense tree of variants where like BA4 mutates and you get a tree of descendants from that, and BA2 mutates and you get a tree of descendants from that. And that's why we end up with these such long names of variants that represent the like the original tree, is that it's like not like one thing going and like completely taking over. It is like a tree, and then all of a sudden you have also the surprise of immunocompromised patients. Out of the blue, you have something that's 20 mutations different, and now that's the thing that then you'll kind of branch off from. So I guess there never has been like a true fixation on like individual sequencing.
Right. Yeah. I think that's been the case with almost all of them, at least early on, like Delta sort of.
Yeah, and I guess, and that's kind of what we've been using EVEscape to do, is say, of the sequences, let's give you a forewarning of which of the many thousands, hundreds of thousands of sequences emerged this month, which one is the one that's likely to take over and dominate. And so that is what we're using EVEscape for.
Yeah, it seems you can predict, because dominant are probably hard to predict the height of.
Yeah.
Because there's a lot more that plays into.
Right, which is like more epidemiological modeling, I would imagine, to try to get at that, like how many people have.
Yeah, it's much more than just antibodies.
Yeah, exactly. Yeah.
General bio.
Yeah.
We got one question to that.
Yes.
Yeah, I just had a question about this natural variability in the broadly neutralizing green patch that you showed, like two or three slides back. Like it was close to the end. So there was a natural variability at the top. One more back, I think.
One more back.
I don't know. It was closer to the end. So you were basically showing the broadly neutralizing target patch, which you said is more conserved. And then at the top it said something like more variable or higher variability. Okay, this is actually good. Yeah. Let's go to that last one. Okay, so this is borne out in the data. The more variable is that what you're saying is subject to sampling, or is it really happening? Is it given the prior data, the all coronavirus data versus the COVID data? Can you say, okay, it's more variable at that top, and so our patch should be at the bottom?
Yeah. So this is based on multiple sequence alignments of coronaviruses and seeing how conserved or different the regions are. But this figure, I don't expect to look very different, even if you were to do it with just SARS-CoV-2.
Okay. So you're trying to be more at the bottom there, at the green?
Yeah, but not too at the bottom, because down here is where the RBD binds the rest of spike. So you want some sweet spot where it's just green, but not blue. That's what we're trying to target.
One more question here.
I have a question back to just how you're calculating the EVEscape score. So when you do the second one, I forget the word, the difference or something, do you have to predict the structures of these different proteins, or are you kind of mapping the sequence onto an existing template, or do you solve all the structures?
So the three components, accessibility does require a one, or in case of the two confirmations, 2 3D structures of spike. From like when you first got that very first sequence in like December 2019 out of Wuhan, you could have alpha folded that. And that turns out that that would have been a pretty good structure. And then you got the conformational structures by March 2020. So fairly early on into the pandemic, we're at the point where it doesn't take you that long if there's like so many groups trying to get structures of this protein because of a pandemic.
So you're using those same structures with the different sequences kind of mapped on, or do you predict new structure?
So accessibility to figure out where an antibody is going to bind, it requires the 2 conformational structures. For our fitness component, where we have all the pandemic sequencing, that's a model of fitness from sequence alone. So when you're looking at all the different past coronaviruses, your like MERS and all your porcine and bat coronaviruses, those are just sequences, those aren't structures.
Okay, and then you mentioned that Deborah Marks thinks that at some point it'll be SARS-CoV-3. What is like the cutoff for calling it a new one?
It's so funny because like where we are in the pandemic, we're as distant from SARS-2 kind of as SARS-1 was. So, but we're even like still calling ourselves Omicron, even though Omicron BA1 and Omicron now are like just as different too. It's kind of crazy. So I think we're gonna have to like get some like drastically different, like some other Sarbecovirus turns into a pandemic to call it SARS-3 because no one's calling it now.
I don't think we should be calling it SARS-3. I think it's still SARS-2. It's just evolving.
And then I have one more question and then I'll give it back, which is, I never heard about this immunocompromised patient with Omicron.
Yeah.
Is that national news?
Yes.
Oh,
I think what initially people had like a few ideas of what it might be. One was that it was evolving in a country that didn't have a lot of sequencing effort, but it seems like most of the evidence is suggesting it's someone who had a chronic infection and then eventually transmitted it to someone.
and so do people do extra monitoring now of like people who are very immune compromised for?
They
new variants?
should, but they don't. And so that's how something like BA2.86, which was again now up to 60 mutations away, was able to occur again in likely immunocompromised patients. Yeah, no extra monitoring.
All right, let me just check if there's any questions on Zoom. Then we can, you guys have really earned your food and beer. Okay, no further questions on Zoom. All right, let's everybody thank Sarah, Noor again. Great talk.